Hello and welcome everyone to this video podcast for Biology 211's lab. This is the second podcast for Module 3. So for Lab 3.2 that we're covering in this podcast, I'm going to talk with you a little bit about the best way to analyze your yeast screen. And then the rest of this time is meant for you to work on your poster and hopefully complete your poster. The next video podcast will go over Lab 4.1, the Drosophila mutants, and there will be something we call star genetics and a genetic screen simulation. And you can read these down here, but these are the due dates that are coming up this week, so please keep those in mind. Again, we're halfway through here for the week. Lab 3.2, analyzing yeast results and working on the poster. Now, when you compare the data from the YPD plates to the stress plate, YPD plus hydrogen peroxide, or the YPG plate, but I really think you'll be able to find something on the hydrogen peroxide plate, you're searching for 3 mutants that grew well on YPD. So the positive control worked. They grew on YPD, but maybe they failed to grow on the stress plate, grew slower on the stress plate, or grew faster on the stress plate. So if you filled out the data sheets from last lab, it'll be easier to kind of go through these data in this way. So for instance, you may see differences between the different mutants and how they grew on YPD plates. That in itself isn't all that interesting, because what we're really interested in is something that had an altered growth on the plates with the stress. So you have to compare the YPD growth plates with the stress plates. So if you look at A3 here, you see it grew on YPD. That's the first criteria. If it doesn't grow on YPD, then we can't trust any of the other data. That positive control has to work. And then we look at A3 on the stress plate and we see no growth. That would be interesting. And we would want to identify what gene A3 represents. Let's look at A5 in this example. A5, we see that it is indeed growing on YPD plates, and we see that it's still growing on the stress plate, but not as well. Just 1 plus compared to 3 pluses. Now let's look at A6. A6, we see 3 pluses here, and we see 3 pluses here. At first, that may not seem all that interesting, and maybe it is, maybe it isn't. But if we look at the pattern, we see that at day 1, there were 2 pluses. Day 1 on just YPD, there was 1 plus. And then it moved to 2 pluses. But on the stress plate, 3 pluses. And it remained at 3 pluses. So it grew at a faster rate on the stress plate. What that means, we don't know, but we know it's different. So it's potentially interesting. So on these series of data, we know we're interested in A3, A5, and A6 here. Now let's look at A8. We see no growth on YPD and no growth on the stress plate. In fact, once you see no growth on YPD, you know immediately you're not interested in that mutant. It could have been an experimental error. It might be that this particular cell culture in A8 has other problems associated with it. But if it doesn't grow under positive control, it doesn't matter what it does here. Even if this was a plus, we still wouldn't be interested in it, because again, the positive control did not work. So are we interested in A1 that we talked about at the beginning here? We see a plus in both of them, just a single plus, so it's clearly growing slow. We see a negative in each of these, day 1s and day 2, for both YPD and stress. So it has a very similar rate for both the positive control and the stress plate. So no, we're not particularly interested in that. Growth is the same on both plates. Now, I went through this on the last podcast, but let me just remind you how you're going to use this A3, A5, and A6 to identify the mutants. On Blackboard, there is a yeast disruption library plate 116 file, and here's a snapshot from that file. That file contains 94 gene mutations in it. But let's just look at these, because in this example, we're only going to look at A3, A5, and A6. Using the row and column, find the ORF. So here we have A3, and then you move down here, and you see this ORF name. For our purposes, you can disregard strain, batch, plate, and record number. It means something, but it's not really that relevant for our purposes here. A5, this is the ORF that we're interested in. And A6, this is the ORF. Remember, ORF means open reading frame. So this is just another way we say gene. Now, I showed a little bit of this on the last podcast, but just to remind you, you're going to take the ORFs identified from that Excel sheet and place it into the search function on the yeastgenome.org database. And after you do that, as you saw in the last podcast, it will pull up a lot of information about that gene. Enough information for you to answer the questions related to this activity and to build your poster. Here's just a reminder in Worksheet 3.2 Part 1. Be sure to write your answer to question 2D in your own words. Now, if this was a face-to-face lab, the analyzing of the data has been occurring throughout the week. You would have been coming in, looking at your data every day. And so this day was primarily meant to work on your poster. So it would be helpful if you would spend the rest of this day that you've devoted for Lab 3.2 to working on your poster. And as you're working with your team, you should be able to get most of it done by this point, and maybe even all of it. So remember, your poster is due Friday at 11:59 p.m. Remember, all team members need to contribute to the poster, and you're only turning in one poster. Also remember to look at the updated poster rubric on Blackboard and not the rubric in the lab module. Once it's completed, you can upload your final poster to a discussion board on Blackboard, and it'll remain on that discussion board so that other classmates can evaluate everyone's posters next week. Looking ahead to the next lab, Lab 4.1, we'll begin our Module 4. It'll be our first and last day for Module 4. In this module, we'll consider the model organism Drosophila melanogaster and work through some genetic screen simulations. All right, that's all I had for this podcast. It's a pretty short one. Remember, most of the time devoted for this lab was meant for you to work on your posters. All right, as you're analyzing your mutants, please let me know if you have any questions, or let your TA know. If you have any questions about your poster, also let us know. But if not, we'll see you in class or lab. Bye.